I think we're gonna go ahead and get started. It is my great pleasure to introduce the guest speaker for today, Leila Wehbe. Leila is an associate professor at the Machine Learning Department and the Neuroscience Institute at Carnegie Mellon University. She is a leading figure in the field of neuro AI, establishing mappings between deep neural networks and the human brain across different domains, including language and vision. She has done her training with other big important figures in this field, a postdoc with Jack Gallant at Berkeley and PhD with Tom Mitchell at CMU. And we are very excited to hear from her today. One little note that I'll say is that this event is co-organized by the NeuroNext initiative, which is the typical host for the talks in this room, which brings together neuroscience researchers from across campus, and the COCO Center, the COCO or Computation and Cognition Center, is a newer center that brings together researchers at the intersection of cognitive science and computation. And if you want to learn more about it and get on our mailing list, etc., feel free to get in touch with me. All right, thank you. Take it away, Leila.
Thank you so much. Hi, everybody. I'm very excited to be here today. And I've had a great day so far. Looking forward to the rest of the day, especially to this presentation, so I can hear back from your feedback or questions. So feel free to interrupt me for any clarification question, and then maybe we can debate more in-depth thing at the end. So I'm going to tell you today about learning representations of complex meaning in the human brain. And at a very general level, what I'm interested in is basically what kind of algorithms we use in order to understand complex meaning in real life. And this is kind of broadly motivated by language processing. So imagine you're sitting on a dinner table with your friends, and you're having some sort of a discussion. And you obviously are going to use language processing in order to do this discussion, but also you're going to use so many other modalities at the same time. Right? You have to even hear and then plan what you're going to say. You have to maybe imagine what other people are saying. You have to plan what you want to say to get them to think a certain thing or feel a certain way. So there's all these other things that you're doing along with language that are just kind of happening at the same time. And the problem is that whenever we study any of those things, we just study them in isolation. So we have not really a good idea of how this language processing thing works. How does it map out to other how does it interact with other people? So I think in order to start understanding more how we make sense of the world in real life, we have to start with really imaging the brain while it responds to complex stimulus that kind of incorporates a lot of multimodal information from the world. So we call this typically a naturalistic experiment. We expose the person and the scanner to a lot of different information. And again, at a very broad level, and this is a goal shared by probably most of the field, nothing specific just to me. Well, the naturalistic experiment is not shared with most of the field, but it's I'm very happy that's becoming more and more common. But really what we want to do, what the main goal is to kind of draw these kinds of brain maps where you have a description of what different brain regions are doing. So this will be not just revealing which areas are active, but also what exactly are those regions doing? So what kind of computation of the input are these regions doing? So as you listen to a sentence, for example, which regions is kind of understanding the audio signal, decomposing it into words, building a sentence structure, understanding grammar, etc. And what are these functions and how do they map to the brain? So more formally, you can think basically of just a region being a function of the input stimulus. There's also a lot of interaction between regions, obviously, but for simplification, you can think of each region as being basically a function of the input stimulus, and we want to kind of reverse engineer what this function is. And when we use a tool like fMRI, the main recording tool, the main unit is the unit of recording. It's called the voxel. It's a volume pixel. And so that's what we're describing here. And as I said, there's not just basically what the regions are doing, but there's also a transfer of information between all these regions. Eventually, we also want to understand that. And there's all sorts of problems in terms of getting there. You have to have the right data that gives you both space and time, etc. So this hopefully eventually we'll be able to get to that kind of an understanding. Now, in order to reach a goal like this, luckily the past decade has really been an opportune time. I'll tell you why. So again, I said we start with complex stimulus that could be multimodal, and we're getting a lot of brain activity from different regions. And now we want to kind of estimate this function, right? We want to estimate each voxel as a function of the stimulus. And typically what we have been doing for a while is to actually simplify this procedure by learning a simple function, g, which is a function of the stimulus. So this phi of the stimulus is this nonlinear transformation of the stimulus that corresponds to our representation of the stimulus, right? And for a long time, the way we've been doing it is by predefining a hypothesis about the stimulus. Like, for example, I think that important thing that the brain is processing is grammatical meaning, and then you can kind of describe the grammar in terms of a vector representation. And then you build a separate linear mapping between those two areas. And this is an area that many people have worked on, including Anya, of course. And the idea is that the reason why you would pick a linear regression model over here is that you don't want to make this mapping very complex, because really this vector representation that you're choosing is going to be like your hypothesis of what the brain is doing at a specific region, and you don't want to mess too much with the hypothesis. And why the past decade has been really exciting is that we have started getting increasingly more complex models for language, vision, and other modalities that we care about. And because we have this has fueled a research movement where we are taking these representations from neural networks, because neural networks are just optimized on really big data sets to solve a task. And in order to solve that task, they just end up learning useful representations. And so instead of like designing these representations by hand, which could be difficult, you just use the activations of the neural network as a stand-in for your vector representation, right? And so like a bit more than a decade ago, this started being used in the domain of vision with by Yemens and DiCarlo, et cetera. And there, the idea was to basically use a neural network for vision as a model or as a source for these representations. Back then, we also started doing this with language models. And so just a recall, a language model is basically an algorithm that estimates the probability of a given word in a context. And so back then, there were like much simpler language models than what we have now. For example, this is a simple recurrent neural network. You have a vector. This vector at the top is like the fixed representation of the word. It doesn't have context in it. It's going to be the same every time you see the same word. And then you also have a representation of everything that happened before the word. This will be like a representation of context. And from that, you can estimate the probability of any word in this particular position without knowing what this word is. So this is supposed to be like a probability distribution. And then once you know what this word is, you can combine it with the context to create the representation for the context in the next position. And from that, also predict the word in the next position. And so, et cetera. You can just keep doing this. And I mean, if you think about it, you're just feeding the model, the text word by word. And this is what we were doing in our experiment. We actually had people sitting in an MEG scanner. So this is a scanner that records the changes in the magnetic field on the top of your brain that are due to neural activity. It has very high temporal resolution. And so we had people read a text word by word. And so we actually thought, okay, so why don't we use these representations from this network to model the brain representation of these people that we are reading from? Recording. And we were able to actually use these representations, and they were very powerful. Like, for example, the context representation is very good at predicting activity even before the word is on the screen, which makes sense because it represents information about the previous words. And then once the word is on the screen, the internal context representation in the brain changes in some sense, and then this older context becomes less and less able to predict the brain activity, whereas this updated context will predict the brain activity. And so this is like kind of a nice tracking of the brain processes using the language network, network's representations. And after this, many other people also started to.
Is it a transformer or is another model?
No, it's just a simpler, much simpler. This is from 2013. It's just a recurrent neural network. So just autoregressive, very simple. And already this does a very good job in kind of predicting the brain activity. There's so much similarity. And this was just one work. And then later on, a lot of other labs started also contributing to this field. And now it's really a subfield in its own, like studying kind of the intersection between AI models and human brain activity. All right. So there's a lot of problems when you do something like this. One main one that gets cited a lot is that the model itself is uninterpretable and has a lot of different types of information in it. And so how can you use it to actually make sense of what's going on in the brain? And I'm going to tell you about 3 different ways we can go beyond this original encoding model stimulus diagram to try to make more sense of this method of mapping between neural networks and brain activity. The first method we're going to talk about is to kind of reverse this process, so construct the original encoding model parameter, and then try to use how well you're predicting brain activity to make some judgments about your vector representation that is coming from a neural network. All right, so the original thing was just this gray lines over here without this arrow. And now the new method is to just kind of use these predictions to kind of judge the vector representation. This will allow us to kind of tell, or the goal we will use this for is to tell what is different about the representation and the human brain. The second method we will talk about is basically end-to-end modeling. So instead of training these networks in a separate step on brain data on the internet, and then mapping it to brain data, what if I directly kind of plug the network, the brain activity at the output of the network, and then try to optimize and find the representation that perfectly predicts the brain activity. So this will be trying to uncover the phi of the stimulus directly instead of doing it in a 2-step process. And we try to see if we train something like that and is successful at predicting brain activity, can we say something about what the output is doing? Can we do more interpretation on this network? And once this network is trained, is it good for something else? Can we transfer what it's learned to AI tasks, for instance? And then we can also look at another way you could use these methods. So once you train your encoding model, could you go back and actually try to optimize basically the firing of different regions in order to kind of create something like a most exciting input, which will allow you to come up with new stimulus that is not in your training distribution that maybe can drive the brain. So this is what we'll be talking about, these 3 different axes. So let's start with the first one. So in this project, we're going to be using MEG recordings, which vary in time. These recordings here, what I'm showing you here is the ability to predict a certain word. This is average over multiple participants. This is why it's a little bit hard to pinpoint specific regions over here. But it's really to see the time. So each word is being shown here for 500 milliseconds. This is supposed to be a diagram of the sensors on the brain. But again, here, this particular results don't make a lot of spatial sense, but just because of the mapping that we used, but at least it makes temporal sense. So we can see here that around 100 milliseconds after word onset, you start being able to predict the brain activity quite well. This is using, this time we are using a transformer model for as a source of representations. And then we're able to predict very well also around 200 to 250 milliseconds, also until 300 milliseconds. And then later on, the ability to predict becomes a bit later. So the predictions of brain activity from these embeddings is very early, and it's kind of sustained. And this is not a new result. This is just us replicating something that people have found using MEG before. It's just going to be a basis for what we want to look at. All right, so now we ask, instead of looking at accuracy, let us look at error. So which word does this model do badly at? So that we're using this model, again, we're using embeddings from a language model to predict brain activity. When we do well, great. But why, when we don't do well, why are we not doing well? Could we extract some general principle of what's going on that, or general observation that explains why we are not predicting certain sentences well? What we're going to do is we're going to look at every word in our test set. And for every word, we're going to label it basically with how much error we do at predicting that particular word. And then we're going to look at sentences once we do that and look at their average error. And we're going to rank our sentences from the most divergent, meaning the most error, having the most error, to the least divergent, meaning having the least error. And we're talking about divergence between the neural network representation and the brain representation. Okay, is that part clear? All right, so now once we have that, we have a set of least divergent sentences, a set of most divergent sentences. And now we want like an automated way to separate between them. And so we use this method by Zhang et al., which uses originally an LLM to come up with hypotheses for why they are different, and then another LLM to do some sort of a permutation test to verify how repeatable those hypotheses are. On top of this, we're also going to do some behavioral tests from those sentences once we get the hypotheses that this LLM is proposing to just verify that these make sense and that people would also agree with these judgments. But the original hypotheses, and by hypothesis, I mean a general rule that differentiates between the most divergent sentences and separates them from the least divergent sentences. That's what I call a hypothesis, and that's what's being verified with human labelers. So these are the top hypotheses that we notice that characterize the sentences that are not well predicted or that are most divergent. And so they kind of cluster across different types of, mainly two different types of sentences or two different types of hypotheses. So we have a lot of things that are related to emotion. So this seems to suggest that sentences that have some emotional components are harder to model in this approach. And another general one is also something to do with like physical reasoning. And then we ask, okay, in order to validate more that this is really what's going on, we ask if we improve this language model we're using. Here, I think we're using GPT-2 because it's really widely used in these kinds of studies. If we improve the ability of the model to do emotion, for instance, and this is really why the model is not doing well, would we actually see an improvement in the ability of this model to align to the brain? That would like kind of validate that emotion is the thing that's differentiating this brain network, this network from the rest of it. And so again, this is the hypothesis. The language models are failing to predict responses due to the limited representation of social or emotional understanding. And the other category was physical word knowledge. And fine-tuning will actually enhance this language model's ability to do alignment. So that would kind of verify that this is really what was missing. And so we're going to do these fine-tuning tasks, in which we pick specific tasks from, those are just typical language model tasks, where we tried, one of the tasks requires some social emotional reasoning, and the other one requires a physical reasoning. And we fine-tune the original model on these tasks, and then we go and we build another encoding model, and we look at how much does our prediction ability change. So here, the top one is the results between the new model that's trained on, fine-tuned on social emotional reasoning and the original baseline model. And the second one is the one that's trained on physical common sense and the original model. And red shows the proportion of sensors on the brain that improve after this. And the blue are sensors that become worse. So a lot of the sensors that become worse actually fall into regions that are not well predicted in the first place. So there might be maybe a little bit of overfitting happening there. The signal is not very reliable in those regions. But for the most important regions, or time points, I mean, like around 100 to maybe 300 milliseconds, we see that in both cases, improving the model's ability to do social reasoning or physical common sense.
This is just the LLM.
No, this is after mapping to the brain.
After mapping to the brain.
Yeah, so this is the ability to predict the brain, and we see that actually improves based on after the fine-tuning. So the model definitely becomes better at the task it has
Can you
refined.
please explain again how you do the mapping with the brain? If you
Yeah, it's basically this way. This way. So we get the brain activity, and then you extract from the network, you extract the embedding. This is your embedding now from the network, and then you build a linear model. Yeah, no, this is the embedding of the input, of the language input.
Okay.
And then you build a linear function. This expresses the brain activity as a function of the input, but the input is expressed in an embedding that comes from your language model. So if the language model embedding is not good and doesn't capture what is relevant for the brain, you're not going to be able to predict the brain very well. And so the theory here is that if we improve the language model's ability to embed emotion, that was a specific question, would it improve its ability to predict brain activity in general? And so we see that it does improve that. And then it also improves, okay. So we do see an improvement. So this is just saying, yes, the whole brain improves. We improve our ability to predict the whole brain, but we can also focus, basically, look at the improvement for social words and non-social words. And we see there is a significant improvement for the social words over the non-social words. So in general, everything improves, but the social words improve also more. And also there is more improvement for the physical word as well in those same kind of early windows of time, so around 200 milliseconds. So for the classical language literature, this is a little bit early of a phenomenon, but we find that when we do these kinds of naturalistic studies and we try to model the brain responses, we always get these like earlier semantic responses than what we expect usually. And we can discuss more why that can be the case. Yeah.
Question on the previous slide. You're showing what we're seeing on the y-axis here is the number of MEG channels.
Yeah.
I was wondering how is it possible that every channel is
Yes, that's a great question. Why is the whole brain improving? It's not really the whole brain. It's again because we're doing this kind of mapping to like it's yeah, I guess the number is not a very useful. Maybe we should do a better plot which kind of captures the effect size. That would be a better way to show this. Maybe something more like this plot. There's a lot of already this MEG signal is smooth and it kind of it's kind of oh sorry here we're only picking the channels that are in the first place predicted by the task. So this is the top channels already, some proportion that have the task in them, but also there's a lot of spatial smoothness. Yeah.
Right, related, well, slightly different version of the same question is, do you have any negative control, which is a fine-tuning task that is not social and physical, that does not change responses or makes the models better? So I'm trying to get at the specificity of social and physical.
Yeah, that's a good question. So our control was more like this, that the kind of the corresponding fine-tuning resulted in more like in those particular words being improved and not the other words. So fine-tuning on social words improves social words more than it does like non-social words. Yeah, exactly. But that's a great question. I mean, a lot of things happen when you fine-tune. You can't just say, yeah, like this is not like the final word and everything. It's just kind of an experiment showing what you could do. Okay, cool. All right, so I'll just tell you very quickly what this is going to be, just like the summary of the talk as we go along. And here so in black we just talk about the methodological contribution, and in red more the scientific insights. So we talked about how to basically study these naturalistic experiments and also how to use basically the divergence between brains and language models as a tool for inquiry. And what we saw is that it seems like language models are representing emotions and physical entities differently than the brain. And then improving the language model on this front makes them more brain. So this was one of the tools I was saying could help with kind of how hard it is to make sense of encoding models and of relating neural networks and brain activity. And the second one I mentioned was actually this brain response optimization or end-to-end modeling, where we take as input a stimulus, and then we're going to optimize a network directly instead of training it on a separate task. Here we're going to study a specific example. It's not going to be a language example. It's going to be a vision example where we're using a large data set of people looking at images in order to train these networks end-to-end on brain activity. And so the idea is that these response optimized models are going to learn the relevant representations from the brain data, and that even given enough data, these models will hopefully be able to approximate these brain processes. And so once they do that, if they're good enough models of the brain, they become like a digital twin or like a in silico brain, and then you can go and interpret these models directly. And you can ask, can this allow us to extract the mechanisms that the brain is using, and whether also these will become better at some AI tasks. So again, these are models that are trained from scratch to predict brain activity. And so in order to do this, we're going to use this really nice large data set that's available online. This is from the University of Minnesota, and it has 10,000 images from each person. Each one of those images, it's 8 subjects. Each one has seen 10,000 images repeated 3 times. So this ends up being 40 hours of scanning for each participant. So they have to come many times over and over to the scanner. So this is like a huge effort, but also it is in 7T, which means the quality of the signal is higher. And there's been a lot of work on the pre-processing to also really drive up the signal to noise ratio. And what results from this data set is actually a very useful, like it's really great. If you are interested in this area and you've not used this data set, I greatly encourage it. It's just really a gold mine. All right. So we're going to use this data set to basically ask whether we can train a model that takes as input a brain image and predicts brain activity, and that's all it knows. It's not going to use any other information. There's no pre-training online. There's no we're not using like ImageNet or Coco or anything in this approach. We're just using those 10,000 brain images. We're going to use like maybe 80% of them for training, and we're going to try to predict a specific brain region. And we're going to try to predict the data of. We're going to use four individuals for training, and then the four remaining ones for testing. So let me just expand on this network. The base of this network is going to be basically just a convnet that kind of mimicking a specific convnet. We do some additional modification. We use rotation equivariant filters because we, again, rely on some assumptions, some heuristics for what could be useful for modeling the brain. The other component is this factorized readout, which is also interesting. So for each voxel in the brain, we don't just estimate linear weights, but we also estimate a receptive field. And so here we're going to try to model high-level regions in the brain, high-level visual regions. So their receptive fields are a bit big, but they still focus more on one region of the image than all of the images at the same time. So this is just basically a filter that tells it where to focus on. And we choose these filters in cross-validation as we are optimizing the network. All right, let me just show you basically how we're going to look at the brain. So we're going to look at this flattened representation of the brain just because it's easier to see the visual system in this way. And I'm just showing you this so that you can orient yourself a little bit. We're looking at regions that fall like in high-level visual cortex around here. Okay, we're going to start with like basically restricting our analysis on specific brain regions just to verify that things are going according to what we think. And so this is a kind of a strong hypothesis to start with, but then we're going to remove this hypothesis and see what happens when we don't have this restriction. So our restriction is going to be to 4 different brain regions. These are visual regions of interest that have been studied for a long time in the domain of vision. And they are identified for each participant based on a separate experiment that the original creators of the dataset have done. Okay, so this is the face area, the visual word form area that is specific for seeing letters. This is a body region that's specific to seeing bodies, and then this one is for places. It's an airspace. So these regions are already identified based on their kind of specialization for these tasks, but this is still a hypothesis. If we don't have this, if we just take these networks and we train a network, we take these regions and we train a network to predict just from looking at images and their paired brain activity, will it also recover the same selectivity? And if it does so, it will be a very strong argument in the favor of this selectivity. All right, so after we train on each region separately, so we are using 4 subjects' data at the same time to train each one of those models. We then validated on held out data. So for our, around 2,000 images, for each image and for each voxel, we have basically the real data for those 2,000 images, and we have the predicted data. We just compute the correlation, and then we identify basically which regions are doing well in this setting. And so this is the pattern of accuracy we get. We're getting up to 0.6 correlation in many brain regions. This is for fMRI. It doesn't sound like a lot, but for fMRI, it's very good. It's really hard to get to that level. I have the plot I can show you later. I don't have it on the screen now. That compares this number to this noise ceiling, which tells you how well you can get, and you get pretty close to the noise ceiling. And remember, this is using this. These are scenes that are very complex. It is not like simple images that we're using. These are complex scenes from the COCO database. That's the input images. They're not like simple. They're really like scenes of everyday life. And also remember that we're training these networks based on only a few thousand images from this data set. When you could do this well also if you use networks trained on ImageNet, or like if you use CLIP or something like that, but those are trained on really so many images. But this is actually just using a small number of images. Okay, so it's great to predict the brain activity. That's a good goal, but that's just the first check. What we really want to do is this interpretation set. So once we have the trained network, we can use methods for interpretation, like, for example, network dissection by David Bao and et al. This method relies on a separate data set this separate data set does not have fMRI with it. It's just a data set of images online. For each one of those images, every single pixel in the image is labeled based on whether it belongs to different concepts. And the concepts fall into like 4 categories. There's object concepts, part concept, materials, and colors. Okay, so each picture, each pixel in this image, in those images online, is going to have these different labels. And so now we're going to do this interpretation work for each image in this new data set, and also for each voxel. Okay, so remember, for each voxel in my experiment, I have the whole model. I can predict the activity for each image in this new data set, and I don't have to have seen the image in training or in the experiment. Okay, and so for that voxel, I can push any image in, and I can take how active the voxel is, and I can look at really the active voxels and then go back and like reverse the operations in the network to find which parts of the input image, really lead to a high activity for this voxel. So in a simpler way, imagine that actually the voxels model is very simple. It's just a linear weight for each pixel in the image. And you can see for that particular image, which multiplication of the pixel value here and the linear weight lead to a positive number. And so this is basically what we're doing here. We're seeing these particular parts of the image lead to positive outputs for the voxel. So basically, this is what activates that particular voxel. And so now, I mean, for this, it's a nice chosen example. It looks like trees here. But what's nice is that we can do this for every concept here. Every concept here has a map like this, a binary map. And you can think of this also as a binary map. And so you can basically do the intersection of these 2 binary map. We use intersection over union. And so we rate now for each voxel, we go through all of the concepts, and we see on average, across all images and all concepts, which concepts really stand out for this voxel. So which concept really intersect very often with what areas of the image this particular voxel is activated by. All right, so this is the interpretation tools. And we can see an example for what happens for the voxels in the face area. So here we're going to look first at the top preferred concept. In this original data set, there were no face, but there were head labels. And you can see overwhelmingly, all of the voxels really care the most about faces. This is not earth-shattering scientifically, but this is done like 100% through just using fMRI to optimize a network. And remember, fMRI is not the cleanest source of data out there. We can also look at basically these regions that are predicted to activate this particular voxel. You can see very clearly this network predicts that this is what the voxel is being activated by. And this is again like what, I mean, this is the part that kind of, I didn't know that, we could do this in now, like we had enough data and we have enough models to really identify this. And we see the same also for the other regions, for example, this visual word form area. So just a thing that there's a lot of overlap between the visual word form area and the face area. That's why you get a lot of head detectors in those areas. But you also get these signboard detectors, even though the original data set had very few images with letters in them at all. And these are the areas, the images in the data set that really activate these voxels. And these are the parts of the data set that activated the most. We also see this with the body area. So person more than head is really what seems to be an activating concept. And for this is an interesting one, the place area we use, we get a lot of window detectors. And I thought about it a lot, and I think one of the reasons is that windows are very rectilinear. This is what these kinds of regions really like. So they have a lot of like horizontal and vertical lines, but also windows tell you about the orientation of an image and also whether you're indoors or outdoors, etc. So the window detector seems to be a nice thing. And also, I think this particular result might also hint maybe at the limitations of such an approach, because for a place area, the notion of place is like kind of a complex thing that maybe cannot be interpreted by looking at just one output node. Maybe you need more like methods that look at interpretation across multiple nodes. All right. But okay, so we did this, and then we were like, okay, so this could be quite cool, but it could also be that actually what we're doing, for example, for the face area, is just training a face detector. It's easy to fit with the face detector. Why would that be? Because maybe the response of the FFA is like always kind of on with some noise when there's a face and off with some noise when there's no face. So is this really interesting, or is it just like we're just training a neural network with noisy data, and 10,000 images, or however, like maybe 8,000 images are enough to do this. And so we said, okay, what if we actually train these models without any pictures of faces or bodies, even of animals? There's no faces in the data set at all. What happens in that case? If you repeat the same thing, what would this network learn about this particular brain region, about the voxels in this brain region? And this is what it learns. Even though it doesn't see any face in training, it maybe sees things that are curvilinear, maybe some things like that maybe look sometimes like faces or something like that. It's able to generalize out of set and still find that these are the images that would excite these voxels the most, and these are the parts of those images that would excite these voxels the most. So it still learns that it is a hat detector. And the same thing happens also for the body regions as well. And so from this, we kind of concluded that these high-level visual regions are not just like an identity thing where like just detecting something and firing them, but they're actually working kind of more continuously, and they're more like domain general filters that really care about specific shapes that are characteristic of their preferred category, basically. One more thing I want to talk about in this case is that these models, as we said, could be actually learning to do something functionally specific that can be transferred. And so we're going to kind of simulate specialized tasks. These tasks are going to be face recognition, letter classification, body pose classification, and room layout estimation. So these are all classification or regression tasks. In order to solve them, we're going to take our original networks, and we're just not going to move the weights anymore. So we're just going to test the ability of these networks to create representations that are useful. And we're just going to put a linear model at the top to solve each one of those. And we're going to train the linear model, the linear layer only. All right, so now we can look at the results. For the face recognition task, we see that the model that's trained on the face area performs the best. And that just improves with training set size. For comparison, this is what an ImageNet trained categorization network does at this task. So we're doing much better than that. This is like 1 million images, right? This is not as good, obviously, as like the state-of-the-art like face recognition network that's trained on a lot of faces, but it's still saying that you learn very good representations for this task from just a few thousand images. We see the same also for the letter discrimination task, where the visual word form area model is the best. The body pose classification task, where really you see the effect with more training data, and the spatial layout estimation task, you also have like kind of a very big gap between the other models and this model. And so we did this kind of also another way of showing that basically there is something that these networks are recovering about the input, what's important for those brain regions, and about the transformation that's important at that particular location in the brain. And as I promised you, if we do this again without having any kind of boundaries set in stone and try to like discover maybe selectivity for a whole big part of the would be the fusiform cortex, encompassing a lot of different visual regions, we still kind of find more or less the same categories. We don't find something like entirely new except for food. So we have the food images, we have a lot of body regions, and head and signboard. So it's not like something completely new comes. It's really just this particular method, maybe because it looks at individual outputs, really recovers the categories we have already identified in the literature. There's no new category. In fact, actually, this food area is something that we and 2 other labs have like kind of found at the same time using that same data set, where there's a region that seems to be really into food. It's like very close to the FFA. We also have a recent, if you're interested, we kind of have a recent review paper or opinion piece that kind of talks about why that could be true. Why could there be a food selectivity region, and why has it like evaded being found until so late, until like 2022. All right, so going back to what we said, so now we've saw that we can use this end-to-end modeling to capture voxel level selectivity. We found basically the same categories we knew about before, but it was like just a stronger proof that that selectivity exists. And we also saw that these high-level visual RIs are kind of more domain general and not, they're active even when their preferred category is not there. We also saw that we can kind of transfer some insight or some representations that can be learned with less data from the brain to AI models. And it seems like I have some time, so I'm happy now I can talk to you about the third approach, which is the most exciting approach. So this is really building on seminal work in the field. These kinds of approaches try to not necessarily use an end-to-end model, but take this approach of kind of taking representation from neural network and then putting a linear layer and predicting a brain region, so taking an encoding model and trying to basically optimize this whole machinery by trying to keep the weights fixed and do gradient descent on the activations to try to generate the input, the image input that would generate the most activity for different voxels or different neurons, and try to say what are those neurons doing based on what is predicted by the model to really activate them. So you can see this is more earlier work, but you can see that these are neurons earlier on, and they don't have, it's not very interpretable, but they have, for example, some neurons, this particular neuron really cares about rectilinear lines. This one is more soft, etc. Those original experiments are not very interpretable because you don't really have a natural image prior. There's been a lot of exciting work also using a natural image prior. For example, this is again in monkeys using, again, it's an earlier approach, which kind of shows, you can see that maybe those look like monkeys. Also very exciting that you can kind of recover these kinds of images. And these are predicted to really activate specific neurons in the monkey. You also see this in work by, Jatan's here, trying to also show that you can actually do this in humans, which is really exciting. And you can really recover these different categories. Later on, there was also work kind of building on Ratan's work, trying to do this with maybe, I think, with the NSD data set as well. And so you can see this, it's really an exciting area of work. And we wanted to just ask whether you could also do this with a diffusion model, and whether the diffusion model could also just generate even larger images, more realistic. And so here, in order to do that, we're going to kind of replace the generative component with a diffusion model. So one portion would be the diffusion model that's going to generate these images. And this is going to be a latent diffusion model that does this denoising process that diffusion models do in the latent space, just becoming a bit more easy to work with. And the second component, which again, building on the previous work, is to use an image computable brain encoder. So here, this, what I mean by image computer brain encoders is something that takes as input an image and then just predicts the brain activity. Let's say we only care about the place regions. Those would be 3 place regions. And I'm just going to focus on that part of the encoder. And the reason why I want it to be image computable is that I can just optimize the whole thing. All right, so this is how it looks like. And I need to use a specific brain encoder. So we found in separate work that actually using something like CLIP is very good for predicting these high-level regions. So this is what we use in that particular work. And then we're going to basically take a gradient that just captures the activity, the average activity in a specific region, like the brain, like the place region, sorry, and then kind of takes that gradient and just goes back to basically the denoising process and just kind of make the denoising process stop, and instead of just like doing what the network would just normally do to sample a normal natural image, it will also make it step towards something that maximizes that specific brain region. And so this is what we get for each case. This is the gradient, and this is the image that's being generated. So this is for the place region, this is for the face region. And you can also see other examples. I really like this example here. This is for the body region. So you see that when you start off, you start with something that looks like a normal image with 3 heads. And then because you're trying to optimize the body region, you got this weird body with so many legs and things. And this is predicted to really activate the brain, the EBA, the body region in the brain. It's unclear that this is true, right? Because maybe the body region just activates more when you have a natural images with a usual number of limbs instead of this like really, like, I don't know, 3 people morphed into one kind of thing. But the prediction from this approach is that it will fire more. So we have to test that prediction. I'll show you a little bit more of our results. So here, for example, for the place voxels, these are not simulated images. These are the original images from the data set. that have the highest activity in the place voxels, the highest activity in the face voxels, and the body regions, the word voxels, etc. So remember those images. We're going to use this as a comparison case when we collect new data. So those images are images from the original data set that lead to the most activity in specific regions. And these are our generated images. They're definitely out of distribution. They're weird, they're extreme, but they kind of correspond semantically to at least what we think these particular regions are doing. So these are like have really exaggerated symbols, like this is an A, has food also. Shoot this. Okay. And this is like more facey. That's already very facey. This is even more facey. This is definitely more limbs. You can see like also there's a sort of, with more examples, you see there's a sort of thinning or like a condensation in semantic space. You really care about this one category with this simulated images. These are the food region, the images that maximize the food regions. And these are our sampled images. You can't really name what food they are, but they really look like food. And so, so this again just means that the diffusion model can be used in the same pipeline that was used before, but just to generate maybe more larger images, let's see. So we still have to test this, right? So in order to test this, the only way you can test these predictions is to collect more data. So we don't have access to the original participants. We have new participants that we recruit. And for these new participants, what we're going to do is we're going to use first a functional localizer. This is what's used to even identify those regions. So this is an experiment that's already exists. That's what has been used the original participant. It's just to know where to look in the brain. The second thing is to basically use those NSD images that we're maximizing from the original experiment. This is going to be our baseline. And then we're going to use these images that we generated that we think maximize the activity more than, they lead to more activity than the original images. All right, so I'll just show you the results in one person. We're still like we just collected the data set, we're still analyzing. The couple of people that we've already processed kind of look the same, so fingers crossed. But basically, the first step is to draw the boundaries of the ROI. We draw these boundaries by hand after computing using the localizer to find the regions that are highly activated by this particular category. We're going to start with body voxels. So again, this is the body, this is the localizer map, this is the activation. This is what you get as a contrast when you compare body to other categories using the original NST images. So you don't get a very clear contrast. And this is what you get when you do the images that we've synthesized, that again, I think are just like so specific to that particular category, then really like really clean up basically the map and almost resemble like the same localizer basically. We can also look at this more quantitatively. So here, each dot here is one of those voxels in the EBA. And it's colored by how strong the original contrast was. And on the x-axis, you see the t-value in the original images. And on the y-axis, you see the t-value for the generated images. And if our prediction is right, it has to all be over the diagonal. And seems to be correct in this for the bodies. For the faces as well, you see this like really above the diagonal pattern by picking those images. The place regions as well have that strong thing. Both place regions we look at. So basically the RSC and the OPA, which is another place region. You also start seeing something that, I mean, we haven't really studied this enough yet, but again, there's like this kind of refinement. It's not as generally distributed anymore. It's really like really special or like very localized in specific regions.
You go back to the first example that you had, the EVA one.
Mm-hmm.
Is it true that the localizer images are having a higher response than the dive images? So I'm looking at the map on the left-hand side, the localizer map and the dive map.
Well, it is. This contrast is not very, like it's clipped at 5 just for viewing. That's why I include this.
I see.
So you can see that this also, a lot of the voxels here have actually a higher, because they're above 10. I think 10 is here maybe. So some voxels do have a higher contrast for the dive images. Yeah, that's a good question. Yeah. Okay.
So this is kind of like MEI, but then for a region that like has single neurons, as in like Wehbe et al, right? So, but then my question is that, okay, so you're optimizing images for each region, like to activate each region. But what if you do like two regions? Like, is it going to be composition of two most exciting images?
Yeah, that's a great question. So the question is about combining multiple regions and maybe compose them. Yeah, we're still working on this. There's been a more recent paper by another group that has tried to do something like that, like maybe maximizing one, minimizing the other, or just minimizing one and trying to interpret this. I think we're still learning how to do this. It's a little bit hard to work with the diffusion models and then do this kind of thing. But I think I can show you an example where, like, for example, we have an, like, it's very interesting to use this composition thing. Let me show you this. Okay, there we go. Okay. Yeah, this one. So for example, here, we basically, so in the original thing I told you, we don't use the caption at all to condition. But here we're going to use the one to maximize the brain region. And then we're going to put a watermelon here in this case. And then you get this like kind of fusion of the two things together. You can try to play a bit around with it. Another way we thought about doing more things is to take two ROIs that fall on the same pathway, try to maximize one and then not the other. So for example, OFA is a region that's kind of lower on the hierarchy than FFA. So you can actually get images that maximize OFA but not FFA, but you cannot do the other way around. That tells you something about the pathway, because whenever you have a face, you're also going to activate the ones early on the pipeline. So I guess I said a lot of things, but the idea is that there's a lot you can do, and we're still trying to uncover how to use those methods for more precise things. And just also to show that it doesn't really work as well everywhere. So for the food region, it's kind of like really like not really there. I mean, they're still activating the way that we would expect, but not more than the original NST images. And also, I mean, it is true that those specific generated images are not maybe as appetizing or interesting looking at the, so maybe there's a more challenge in generating the food images. So, okay. So as conclusion for this case, most exciting inputs can actually reveal our level selectivity. We actually use them more in the paper, where we try to even separate, like discover something new and look at within the same ROI and try to see if like different parts of the same ROIs have different selectivity. And we do have some success doing that. And the idea here is that we kind of validate this experiment also with new data, where it means that, you know, like we can kind of use these out of distribution images to drive the project, to drive the brain activity. Okay, I'm done. I'm just going to tell you about this ongoing project that we're doing right now, where we're collecting a lot of data. This is in collaboration with Alex Huth. Alex is doing the fMRI part. We're doing the MEG part. We're trying to get a very large language data set, which we will share hopefully soon, as soon as it's, you know, physically, as soon as it's done. We're also going to try to do the same thing with this, where we're trying to train language models, either pre-trained or not, to predict the brain activity, and trying to say whether this like allows us to distinguish between existing neurobiology of language theories, but also trying to see if like different parts of the brain really resemble more maybe different language tasks. I'm not sure how easy this is going to be, but we're also, of course, going to share the data, a lot of useful software packages, and also the models we train. So please stay tuned, and you know, let me know if you would like to be part of the early release at some point whenever we do that. And with that, I would like to basically thank all of my collaborators, and thank you for listening.
I am new in this field, and so I'll try to understand. So from what I understood, the transformer, that the key element, the core of LLM, what he does to the self-attrition layer, each token is like a particle that it moves on a high-dimensional sphere, and basically, you know, the transformers, you know, can learn the attraction forces between the tokens in such a way, you know, they can cluster depending upon and learning patterns that they see on the huge amount of data. And so basically, the learning rules, the grammar, the semantic, they're all encoded in the clustering of these particles. Yeah. So that's what basically, that's what it is, the transformers. So now, how can we talk? I mean, when you talk about being LLM being emotional, I mean, it can never be like a human emotion. So it doesn't have consequences, right? So now, when you do these patterns that you're able to identify in the brain, can we say that those are kind of nonlinear mapping between the clustering that is already encoded in the LLM and these clusters that we have in the brain? So do we have
one? Yeah, definitely. This is by clustering, I guess the way people think about it is that words are like words that are used in a similar context in language have similar meaning, and you can also see that they have similar brain responses, and this is what is actually fueling this ability to predict the brain activity from the input. Yeah, definitely, and from these different things.
Can we learn? Can we decode what are the grammar rules that are learned by the LLM?
Yeah, I mean, this is one of the reasons for wanting to like do this kind of big data set, so you could maybe try to learn the rules.
How are you going to do that? Because the information.
Let's maybe make sure that other people can also ask questions.
Okay.
And then you can take it offline.
Okay.
Yeah. Okay. Just wanted to make sure that people who want to ask questions as well.
Any other questions?
Can I venture one then? So you showed that food one where it was like on the unity slope. So at that point, like, how will we be able to diagnose if the problem is with the generator or the encoder itself?
Yeah, that's a good question. Yeah. Well, not very. Like I don't have an answer that solves the problem, but you're totally right. It could be done. It could be too many things.
Yeah. Cool stuff.
Yeah, thanks.
I'm just curious. Have you tried to do the visual declaration experiment for words? Because like words, you learn that, but the data you train is on adults. So actually, so it might have like already have the words information.
Yeah, we didn't do this because there was like less images with words. But yeah, I mean, yeah, that's a good question. Yeah, like there's always more complicated things happening with the word. Yeah. But yeah, I think this is one of the experiments we have on our list for like the revision for the paper. So and hopefully it will show up in the paper. Yeah.
On that topic, so how do you reconcile your domain general account of the selectivity with the fact that you only find selectivity for like 4 or 5 things?
Yeah.
Right. What's the
Yeah, it is a great question. I don't know. I'm still thinking about it. I think it does matter when like these are just like a more complex version of the original localizer experiments, basically. I feel like you're still looking for the peak, you're still looking what the top activity is, but probably if you have a truly more distributed way to look at this, I mean, this is distributed in the way we look at we model all the outputs. We're not trying to interpret the pattern maybe, or we're not trying to, it's also hard because I don't know what like a multivoxel pattern means in the first place. Yeah, it's hard to reconcile it. I agree, but yeah, it's definitely in there. We're using the brain to recognize lots of objects. Yeah.
With auditory signals and compared that to how the visual signals were getting a response. So if you're describing like the doctor told the patient she was ready to see her, what if you were looking instead at a stimulus that you're getting from your GPS and how that might be able to direct people to respond better to redistribute people across roadways or something like that? Would that affect the same parts of the brain, or does that look different? And have you all looked at anything like that?
So we definitely, so this data we're collecting is going to be audio. We haven't done the kind of maximizing experiments for audio. Some people have. I don't know that that's a very interesting, I think this is the one of the interesting future goals. The motivation why I'm insisting that this is not just about discovering what a brain region does, but provide a new way to explore, like a new, like a prediction that, oh, this stimulus can do this. And that kind of application would be great. Like, how do you design an audio input that actually people listen to? That
You
could
could
be.
cross over, because with the GPS, you had the audio and you'd have the simplistic visual input.
Mhm.
You could try to look at the three, I don't know.
Yeah, like just yeah, I guess the flavor you're saying is like trying to be more interventional, trying to like design new ways of, yeah, that would be really a dream to work on something.
Thank you.
Sure.
All right, thank you so much.
Thank you.